I do find this is one of the units that it's really, really important. Any of you who've done clinical, if you've done your clinical rotation at all, you know that you're going to have a ton of diabetic patients. So whether you're on a medicine unit or a cardiac unit or even in Emerge, whatever, there's just constantly insulin. Like I plan my whole day around having to give out insulin before meals, check on people after they eat. And it took me a while to kind of get used to that scheduling. But anyway, it's just a huge unit. But then I find people, it's pretty easy, but then I find people like forget in between when you take it and when you go to work, like how insulin works. So I'm going to go over like just the basics. Hopefully you guys, I did, Kieran, how was this test? Do you remember? Because it's like, it's the 2 diabetes, the obesity and metabolic syndrome, and then I think the mental health stuff is on that test too as well, right?
Yeah.
Okay. Do you remember how it was?
So this one wasn't bad. I feel like it was the questions that they asked, I feel like the answers were kind of obvious.
Good.
Which is good. Usually it's not like that. But I feel like it's hard to wrap your head around diabetes sometimes. It can
Yeah.
be a little confusing, but like I feel like if you go through it slowly and like just connect some things together, it kind of makes sense. Like don't focus on the specific stuff because they're not going to ask you that. Focus like symptoms and what's the difference between type 1 and type 2, and knowing the stuff like that is I think important, but not, and also knowing like how it has an effect on like the kidneys and all of that. So
right. Well, and I think again, they're going to ask you nursing questions, so you don't need to go into like Shireen put up a lot of stuff about like amylin and somatostatin and all these things. I think just having like a basic understanding of those is going to be okay. So let's start with insulin. So you guys know that your body makes insulin on its own. It's endogenous. Anytime you see endogenous, that means the body makes it on its own, right? So sorry, I just want to get rid of this here. So it's produced in your beta cells inside of your pancreas. You will see a lot of references to islet cells, beta cells and whatever in your test. Just make sure you understand that that's where your insulin is being made. It's continuously being released in these kind of small pulsatile increments. That's your basal rate. And obviously it decreases your blood glucose. This entire like unit or what have you is focused on how much glucose is in your blood and how or if your body is able to use the glucose is there. So doesn't really matter, but an average of 40 to 50 units a day. This is the kind of stuff I wouldn't worry about memorizing. It's just good to know. So most of your, remember when we did, oh yeah, you guys did kidney. Yes, you did. How your nephron was like your kind of active part of the kidney. It's kind of the same with the pancreas. Pancreas is made up of these kind of small clusters of cells. They're called acini. Doesn't really matter. Inside of those or around those kind of, you have these alpha, beta, delta cells. An islet of Lange, if you see this is a kind of an islet cell kind of here, but it's got like the alpha and beta cells inside of it. I do think knowing that that pro-insulin is released from the beta cell first. And pro-insulin is just this, it's a larger molecule. It's made up of an A polypeptide chain and a B polypeptide chain. And it's, if you see here, I kind of tried to make it a little more visual. It's attached by this other one, this C peptide chain. And what happens is the ribosomes, and always zomes is going to be an enzyme, right? And enzymes generally break things up. So there are these ribosomes in the beta cells that cleave or separate or chop or however you want to imagine it, the C chain from the molecule. So it separates the big molecule into these two smaller three really. You've got your C chain molecule, and then these two insulin molecules. And so this is really cool. This C chain that's left over, it actually just stays in the blood, and so it can be used to, like if we do a blood test, you can actually see how much of that little molecule is left inside of the blood, and that tells us how much insulin is actually being released from those beta cells, right, in order to be turned into proinsulin, in order to be turned into insulin. Basically, insulin, the whole point of insulin is to help your body regulate and use glucose. Okay, so who cares what, well, let's talk about glucose really quickly, because I find this gets kind of glossed over, and you know, if we're not sure, if we don't remember our grade 11 or grade 12 biology, it's kind of confusing. So glucose, all the cells need glucose for cellular respiration and metabolism. If you remember, you've got this whole, well, actually it's right here. If your body has to make all of this energy, right? And it goes through all of these kind of steps. The first step is glycolysis, and then you have that lovely Krebs cycle, if you remember that, and then the electron transfer, that oxidative phosphorylation. Don't worry, you don't have to know any of that. But the point is that it starts off with basically with glucose and oxygen, and ends up with carbon dioxide, water, and this should be 34, not 36, 34 ATP, that's your energy molecule, or the way we measure energy. So this is the whole point. Without this glucose, our body cannot perform this kind of respiration and create energy, right? It's the same when we were talking about the body going into anabolism and anaerobic metabolism, right? Because it doesn't have oxygen. So you need oxygen and glucose just to complete this equation and get energy. That's the whole point. Now, unfortunately, while like oxygen, water, carbon dioxide, all of those other things can pass in and out of a cell through osmosis, glucose can't, of course. And so we need insulin to kind of help the glucose get into the cell. So it's really important to understand that when you're measuring glucose, so when you do a CBG or blood glucose or what have you, you're measuring the amount of glucose in the blood, right? And so what we want that glucose to do, the glucose that is in the blood, is we want it to be used either by the cells, or we want it to be stored in the liver, and then so that it can be used later when maybe we don't have as much glucose kicking around in the blood, right? So a lot of people kind of look at this as like a key and lock. I don't know, that I find that metaphor kind of breaks down really quickly. But if you look at this, if this is your cell right here, and you have this glucose, these little glucose molecules here that are trying to get into the cell, but they can't get in, because actually this glucose receptor right here, which it's a GLUT4 receptor, because it's on this. It looks like we're going to pretend it's a fat cell, because that's what it looks like. So this receptor actually is located below the surface of the cell. And the only way that receptor can find its way up into the cell where it's supposed to be is if insulin attaches to the insulin receptor on the cell. When that happens, it triggers this whole cascade of enzymes ends some cleave this, some do, it doesn't really matter. It's just a big cascade that eventually ends up kind of pushing that glucose channel or glucose receptor, whatever have you, kind of halfway out or into the cellular membrane. And that way, if you look over here, that way now the glucose can open, the channel opens, and the glucose can enter the cell. So if you want to think of insulin as the key that unlocks kind of the door of the cell or whatever to let glucose in, that can be helpful. So if let me just see here. Let's go to these receptors really quickly first. So we're talking about these receptors, right? These ones right here. These are glucose receptors on the cell. There's a bunch of different kinds. Really what's important for your test to know is this one, this Gluc4, because it's the one that requires insulin. Actually, these first two, Gluc1 and Gluc2, they're in many tissues in the body, most especially in your nerve cells. Really important for neurofunction. Doesn't require insulin. So this Gluc1 receptor on, say it's on a nerve cell, and I'm not even going to try to draw a nerve cell. It's on the nerve cell. It's actually already popped up in the membrane. Doesn't need insulin to kind of help it get up, right? Same thing with Gluc2. Gluc2 is mostly found in your beta and liver cells. It's not, these ones aren't, when we're talking about insulin and glucose receptors and the problems between, we're mostly talking about Gluc4 because these are the ones that require insulin. These two do not require insulin. The receptors are already at the surface. Gluc4 receptors are located on skeletal muscle and adipose tissue. So fat and muscle, that's where those receptors are located that need the insulin. And we've talked a little bit about, well, we'll talk more. Sorry, it's unable to transfer that glucose through that cell membrane without insulin. That's where this whole thing is happening. So that's really important to remember. Gluc4 is located on skeletal muscle and adipose tissue. I don't know if that will come up, but it's definitely come up on some of my tests. So this is where things get a little bit confusing. There's a lot of these words here, glycolysis, glycogenesis, what have you. Some people get them right away, no problem, and don't feel bad if you don't. I usually have to go over them a few times. So what is insulin doing? Insulin, it's at its very base, is helping the body use glucose, right? In order to turn the fats and proteins and carbs that you eat into energy or store it as glycogen. So if insulin is aiming to use glucose, right, it wants to decrease the amount of glucose that's in the blood. It wants to put that glucose into a cell or into a million cells, or it wants to put it together into glycogen and put it and store it in the liver for later use. So if you remember that, all of these things make so much more sense. So let's start with glycolysis. Glycolysis is this process of, if we look over here, remember we were talking about cellular respiration and how it has these like 3 stages. The first one was glycolysis, right? It's actually what turns glucose, there's a couple of other things, turns it into this pyruvate. If you don't remember, it doesn't matter. The important thing to know is that it's kind of what kicks off this process of turning glucose and oxygen into energy. So glycolysis is really, really important. So glycolysis is using glucose and turning it into energy. So yeah, so that's what insulin is going to support that. That's what insulin wants to do, right? It also will increase, glycogenesis. Glycogen, okay, is a molecule that is made of glucose molecules, and that can be put together and then stored in the liver. So the term for when that happens is glycogenesis. So if you just look at the word, you guys know we've talked a million times. Genesis is the building of, or the making of, or the beginning of. And in this case, we're talking about glycogen. If you look over here, if you imagine that glucose looks like this, your glycogen, if you imagine the glycogen a kind of a string of glucose molecules, they're kind of strung together and then stored as glycogen in the liver. Why is that? Because insulin wants to get the glucose out of the blood and either into the cells or into the liver, right? And then you have an increase in the glucose uptake into fat, right? Again, it's taking the glucose out of the blood and putting it into your fat. What does the fat do with it? It makes glycerol. You remember we looked at those kind of triglycerides, and it had that glycerol, and then it had those 3 fatty acids kind of hanging off of it. This right here needs glucose to make that glycerol molecule, right? And this is how it's storing kind of that glucose or that energy, not just, well, it's storing it in our fat cells. Remember we talked about how glucose is, or how the body will break down fat for energy if it doesn't have glucose. So again, this is another instance where insulin is helping get glucose out of the blood. This time it's going into fat. So here we've got it used for energy. Here we've got it turning into glycogen, being stored in the liver. And now we've got it being taken into the fat and turned into, basically into triglycerides and fatty acids. And so there's a word for that as well. So you've got lipogenesis, super, super easy. It's the making of, or the beginning of fat, right? Lipo, fat. So if you see these words on the test, just think it through. Lipogenesis, we're making fat. We're creating fat. We're creating a triglyceride molecule out of glucose. So what's happening to the glucose? It's coming out of the blood and being used. Anytime you see some process where glucose is being used in the body, you know that insulin is helping to do that. And so, because you're definitely going to get a question that's going to say, you're going to have to pinpoint that insulin does this, this, and this, and doesn't do this, this, and this, right? So you just have to kind of think it through instead of trying to memorize all these words, right? It's also going to increase the uptake in muscle, because again, those GLUT4 receptors are located in muscle and fat. What does the muscle cell use it for? Energy. And then it's going to also uptake amino acids. And what this does is it synthesizes protein. Protein's used for a ton of stuff in our body, right? Like most of what we're talking about are made of proteins anyway. But again, uptake into muscle and uptake into amino acid, that's all pulling the glucose out of the blood and using it somewhere, right? So it makes sense. So when we don't have, this is probably, so when we have a lot of insulin in our body, all the other methods of making glucose or energy are going to slow down. So if you just look over here, you remember that if you don't have glucose in your body, what does your body do? First thing it's going to do is it's going to break down fats. So it's going to break down these triglycerides here, and they're going to break it into fatty acids and glycerol, and and turn that into energy. Ultimately, once that's kind of depleted, it will start to break down muscle for the amino acids. Remember, we took those amino acids here and we turned them into protein. right? Now we're going to take the protein that's in the muscle and we're going to break it down and we're going to turn it into amino acid, which can then be used to produce energy as well, okay? So that's kind of what the body does when it doesn't have glucose or when it can't use the glucose that it has, right? And so if you have a lot of insulin in your body and all of your glucose or a lot of your glucose is being used, then you don't need any of those things to be happening. So it's really, it makes sense that you're going to have a decrease in gluconeogenesis. Gluconeogenesis is glucose, new, and making of or beginning of. So this is the process of making new glucose. And you're going to have a decrease in glycogenolysis. So that's really easy though, right? It's glycogen that you're breaking up. You remember I said that we store glucose, if you imagine, you kind of string it together into this glycogen, this large glycogen model, we store it in the liver. If we have lots of insulin in the body and all of our glucose is being used, then we don't need to break those down and release them into the system. They can stay there. We don't need the liver, we don't need that help, right? And then again, we have a decrease in lipolysis. And this is the same thing. It's that breaking up of fat or triglycerides to release that glycerol and fatty acid, and a decrease in protein breakdown. So if you just kind of look at it as, okay, I have lots of insulin, so all of my glucose is being used. I don't have to resort to any of these other measures. And you know that anything that makes glucose or anything where the end product is glucose, whether it's gluconeogenesis, glycogenolysis, or lipolysis, all of those are going to end up making more glucose. You don't need that because you have so much insulin. You're already all of that glucose is already being used. So instead of just trying to kind of memorize all of these words, this is a really, really important kind of section. That's why I'm like hammering it home. But if you go over this a few times, make sure you understand what all these words mean, and then just think to yourself, you know, this is what insulin does. So if whatever the word is, if it's using using glucose, it's probably something that insulin does. If it's creating glucose, it's probably something that will slow down when we have insulin in the body. Whew. Okay, so that's kind of like it's kind of like the backbone of everything we're going to talk about. So before I go on, is there any confusion about that? And seriously, just ask now, even if you need me to repeat. I know I said it twice, but if there's anything you're confused on about any of this, if I didn't explain it well enough, please ask and I'll go over it again. It's totally fine to do that. Yeah, I know you're good, Saeed. You probably understand it better than me. Just let me pull up okay, so everyone's okay with that. Just making sure. Okay, I did all of that. We did that. Let's talk about counter-regulatory hormones then. So these are counter-regulatory hormones are hormones that work against insulin. So because insulin is a regulator or regulates glucose, counter-regulatory are the things that work against insulin, or they increase glucose. So if you look back down here at this, these counter-regulatory hormones are going to do all of these things, and they're going to slow down these things. They work against that insulin, right? And so your biggest one, really most important one, is your glucagon. We'll talk a lot about it, but just kind of an overview. Glucagon is this kind of polypeptide molecule. This time it's secreted by the alpha cells and the same islets, the same, I think I have a drawing down here, kind of that same islet here, right? But then instead of releasing from the beta cells, it's releasing from the alpha cells. Definitely something you want to know. Glucagon, you know, it's something that glucagon's goal basically is to keep glucose in the blood, to make sure that there is enough glucose there in the blood to be used between meals or if you're fasting. Just like insulin, it's going to travel through the portal vein to the liver and exert its main action is in the liver, only instead of decreasing blood glucose, it's going to increase blood glucose. So it's going to initiate, it's going to start glycogenolysis, right? It's going to break up glycogen into glucose. It's going to stimulate amino acid conversion, so it's going to, or gluconeogenesis. It's going to turn amino acids into glucose. It's going to, yeah. So that kind of, this glucagon is working for glucose in the blood, right? Whereas insulin is working against that. So I know I'm repeating myself. It's just so important. Then we have these other kind of hormones as well, or some of them are, we've seen some of them before. For example, epinephrine, right? You know, epinephrine is secreted by the adrenal medulla. This is the same hormone we've talked about when we talk about your sympathetic nervous system response or your fight or flight response. Again, epinephrine is going to do the opposite of insulin. It wants to keep or have a good amount of glucose in the blood to be used. And that's because if you think of it as like I'm in a fight or flight, I need as much glucose as I can to feed my muscles so that I can run or whatever. I always think of a bear chasing me for some reason. But like if you think of it like that, that's kind of epinephrine's goal, right? Is to get you out of a sticky situation. It's a fight or flight. So it's going to stimulate glycogenolysis, right? It's going to stimulate that breaking up of glycogen in the liver because the body needs more glucose for energy to survive whatever it is that it's running from or fighting against, right? And epinephrine actually inhibits insulin release. And that's because we don't want to break down, we don't want the glucose leaving the blood, right? We want to keep it in the blood. Epinephrine kind of, again, like the glucagon, kind of like wants to like hoard that glucose in the blood. What that does, you're like, well, that doesn't make sense because if we need that glucose to like run away from a bear, how if epinephrine's keeping in the blood, how are we using it? The thing is that it basically what it does is it increases the availability of that glucose for the brain and the nervous system. So it's a fight or flight response, right? It's going to shunt all of the energy to the main or the major systems. So there's less insulin in the blood, less of that glucose is being absorbed by other systems, and it's being shunted straight to the brain and the heart where the body needs it, okay? Then we've got these, I don't know if you guys focused on glucocorticoids as much, really, really similar, secreted by the adrenal, they're adrenal gland or adrenal hormones, right? So it's going to do all of the same things that epinephrine does. It's interesting though, because if you look at this, you know, cortisol, glucocorticoids are basically when we give corticosteroids, right? It's basically we're trying to kick that kind of system into gear. We're kind of synthesizing that fight or flight by giving, or not fight or flight, but a bunch of different things. Usually we're giving corticosteroids for its anti-inflammatory purposes, right? But it's actually going to kick in some of this stuff too. And that's why when we give corticosteroids, we tell people they're going to find probably an increase in their glucose levels, in their blood glucose, because glucocorticoid or corticosteroids or cortisol, they're counter-regulatory hormones. So they want to hoard that blood. They want to hoard that glucose in the blood. Growth hormone is really, really similar. It's just secreted by the pituitary gland. It's basically like the body's reaction to fasting or starvation, right? It wants to increase protein synthesis, decrease the effects of insulin to keep glucose in the blood for use, decreases cellular uptake of glucose. It just wants to hoard that glucose in the blood. That's what all of these guys want to do. Just wants to like pull all that glucose in. And again, if we don't have insulin, if we're hoarding, if we're lowering the output or whatever of insulin, we are going to have a lot less of this happening and a lot less glucose getting into the cells. So really actually, let's go over here really quickly. I really like these I feel like this really helps break it down. So let's say your body is hypoglycemic, meaning, again, when we talk about hypoglycemia and hyperglycemia, we're talking about the amount of glucose in the blood, right? So you are hypoglycemic, meaning you don't have a lot of glucose in your blood. Immediately, the body is going to kick in, and it's basically a feedback system, which I'll show you in a minute, but it's going to kick in, it's going to release those counter-regulatory hormones, because all of those counter-regulatory hormones are increasing the glucose in your blood, right? So it's going to release glucagon. Glucagon is going to increase, glycogenolysis. Some of these words, they just stick. Glycogenolysis, so breaking up all of that glycogen in the liver and turning it into glucose, right? Your body's it's going to also release epinephrine, which is going to increase your lipolysis, so basically pulling glucose out of fat, breaking down your fat and making it into glucose. Now, always remembering that you're going to release ketones when that happens. There shouldn't be a Y there, sorry. And then it's going to release these other counter-regulatory hormones as well, growth hormone and cortisol. And they all basically do very, very similar things until you end up with more glucose in your system. Your body kind of returns to its homeostasis and everything kind of goes back to normal. So really, really quickly before, these are not... I mean, Karen, oh, you know what, you guys, sorry. You didn't have a lot of this in your notes from last year anyway, so you wouldn't have gotten it on the test. But these are kind of some of the other hormones or whatever that are released from those islet cells. Sorry, I'm just going to check which glucose producing, sorry, Darla's asking. Which glucose-producing processes do growth hormone and glucocorticoids trigger? So glucocorticoids are going to do very, very similar to epinephrine. But the growth hormone, if you look here, and cortisol, if you look here, the growth hormone is going to decrease insulin sensitivity, meaning all of this, these cells that normally need that insulin to kind of get that receptor up and out through the membrane, they're not going to be as sensitive to this insulin. They're not going to, literally, I just look at it as like they don't sense it, they don't see it, they don't hear it, they don't know it's there, so they don't use it. And when you have a decrease in sensitivity, that means all these glucose molecules are going to stay in the blood. Basically, it's decreasing. So growth hormone is decreasing the glucose that's being used by the cells, so there's more in the blood. And then cortisol specifically is going to increase that protein breakdown, and so that will release amino acids, and then basically you can make brand new glucose from that and some other things in the liver, and that will increase your glucose as well. You don't necessarily need to have all of these pathways memorized. I just drew it out because it makes more sense to me to see, like, well, how is that doing that? The chances of you having to know specifically for a test are pretty thin, but if you get one, all you have to do is think it through. You know it's a counter-regulatory hormone, so it's going to be trying to hoard hoard glucose in the blood. So just pick whichever ones are going to do that. For example, lipolysis is going to increase the amount of glucose in the blood because you're breaking down the fats and releasing the glycerol and the fatty acids, right? So you just have to kind of think it through. It won't be as complicated as like knowing, you know, what each one does kind of specifically. Or if it is, it'll be very like, you should be able to kind of pick it out, I guess is what I mean, just by logic, with logic. So again, we've got this pancreas, these acini and this islet cell, and inside of the islet cell we have alpha and beta and delta cells, and we already know that the beta cells are releasing insulin, and we know that the alpha cells are releasing glucagon. And we talked about glucagon being a counter-regulatory hormone. So it's going to hoard glucose in the blood. So it's going to do these things. If you walk yourself through it, it'll make sense. This amylin is also secreted by the beta cells, kind of along with insulin, right? It decreases gastric emptying. It decreases appetite. What's important for this unit is that it blunts the secretion of glucagon. So if glucagon is a counter-regulatory hormone and amylin blunts that or stops it or makes less of it secrete or whatever come into the system, then what's going to happen? If you have less counter-regulatory hormone, then the insulin is able to do its job, right? So you just kind of have to kind of think through it like that. Somatostatin, again, I'm not even sure. I think they just need to get like as much of the pancreatic stuff in as they can, because these have more to do with like hunger and satiety, I guess. Anyway, somatostatin is secreted by the delta cells. So you've got alpha, beta, delta. The somatostatin is actually, most of your body's somatostatin is located in your GI system. It will decrease kind of those hormones that are released in the GI system. It will decrease insulin and glucagon. So to me, it's just like, okay, so it kind of cancels itself out. I'm not too worried about that. Decreases motility in the stomach. So really, you're slowing down the GI tract, slowing down, you know, what the GI tract is doing. When it talks about secretion, secretion and absorption in the GI tract, you know, they have those, I just want to make sure I don't miss it, gastrin, motilin, I can't remember, that CCK we talked about last week. All of those things are kind of things that the GI tract is secreting, and have a lot to do with digestion, and also with with insulin, but they're not like, as far as I know and how I've always thought of it, is they don't have a huge influence on like this kind of relationship between insulin and glucagon, but they do support one or the other. So amylin, for example, will support insulin. If you look over here, you're going to find that these other hormones, they're called incretin hormones because, again, they support insulin as well. So these incretin hormones, these are hormones that are secreted by the small intestine after eating. You have this glucagon-like peptide, or GLP. We did talk about that last week, right? Who cares that it's released from the L cells? It's definitely not something you have to memorize. In the distal small bowel, which is the ileum. And then you have this glucose-dependent insulinotropic polypeptide, or GIP. Again, we don't care that it's released by the K cells for our purposes. And it's in the upper, I think the slide said upper gut, basically mostly in your jejunum. The important thing to know about those two hormones Again, anytime you see the word incretin, it means that it's supporting or stimulating insulin release, and usually a decrease in glucagon. Because remember, insulin and glucagon are working against each other. So most of these hormones that we're talking about, this amylin, GLP, GIP, they're working on the side of insulin. So if you get, bogged up in a test and you're not really sure, just remember those ones are working with insulin. The only ones that are working against are the ones that we listed up here. And we already went through like how they do that. So this is kind of the main like thing that you have to understand, is this relationship between insulin and glucagon. This antagonistic kind of relationship they have, because they have opposite effects. It's very, very tightly regulated kind of feedback loop. I know sometimes these feedback loops can be a bit much, but we're going to go through, really, this one's not bad at all. So if you look in the middle here, this is when we have homeostasis. This is our perfect, lovely glucose level. I'm sorry, I couldn't find a Canadian one, but whatever. And so let's say the first thing that happens is that we eat a big meal and our blood glucose begins to rise. So now we're a little bit hyperglycemic. The body is going to stimulate the pancreas to secrete insulin. So right from the B cells, the beta cells, the insulin is released. And we know it's like in those chains and all that stuff we talked about earlier, but this is the basic concept. Some of that insulin is going to be used. It's going to be taken up into those it's going to be used to help take up the glucose in the muscle cells and fat cells, like we talked about. And then some of it is going to go to the liver, and it's going to be turned into glycogen, and it's going to be stored there. And then, so, when you're hyperglycemic, it's not just the insulin dependent cells that are going to take up glucose, right? All the cells that need glucose are going to take it up. They're going to use it. And then eventually you get to the point where it's pulled enough of that glucose out of the blood and into the cells, or it's stored enough inside of the liver that your blood glucose has declined enough that it's back to your homeostatic balance. So let's say now, instead of the stimulus being hyperglycemia, it's hypoglycemia. You haven't eaten in a really long time, you're hypoglycemic. It's going to trigger now the pancreas to, instead of releasing insulin from the beta cells, it's going to release glucagon from the alpha cells. Because we want to get some glucose back into our blood. And this glucagon hoards glucose in the blood. And so the body releases glucagon. And really what it does is it sends the message kind of to the liver to break down that glycogen. Remember that glycogen we stored up here? It's going to break it down now, and it's going to release glucose into the blood and release enough glucose to bring it back into the homeostatic balance. Now, glucagon does a couple of other things, but this is like the basic kind of feedback loop. If you have a good grasp on this, and you understand what all of those, fancy words are, glycogenolysis and whatever, then you'll be absolutely fine to be able to answer the questions. There was something on your slides, one of your slides, that said insulin was anabolic. And so just in case you need to know that, I guess, this is where it would have been helpful to have done this last week. But basically, you have these two types of metabolism, right? You have anabolism and catabolism, and I don't care. Some people say catabolism and I don't care. I'm just going to say it the way I say it. One of them creates energy, one of them uses energy, but in this case, just think of it as anabolism is going to take smaller molecules and turn them into one large molecule. That sounds kind of familiar, right? Because we talked about how that glucose molecule, when we turn it into glycogen, it really just takes a bunch of those glucose molecules and ties them all together and makes a bigger molecule. And that's why, and we know that insulin is what does that, right? Because it wants to store that glucose as glycogen in the liver. And so that's why we would consider insulin to be an anabolic hormone, and then your glucagon then of course would be your catabolic, because it does the opposite. It takes the large molecule and breaks it down into the smaller molecules. So just in case you get stuck, you have a question on that, just look it over, make sure you kind of have an idea of which is which. Okay, so I'm gonna need, so Darla, you had said that Shireen went over a lot this kind of like pattern of insulin release in the body, like that basal bolus pattern. But the only slide that I had only had like that biphasic, the biphasic release. Is this what she talked? Because so I've always, the way I have always understood it is this pattern here, but I didn't see this anywhere in your slides, but I just don't want to cover it wrong for you guys. So what exactly did you cover when you talked about this kind of pattern of insulin release in the body? Sorry to hopefully you're around. Nope. Anyone? Darla? Maybe not. Okay. So I don't really know. Oh, it's similar to that. Okay. Sorry, did I freak you out? I'm running. To me, this is what I know. This is what makes sense. You have the way your insulin is released in your body, and this is kind of what we try to mimic when we give exogenous insulin. Yeah, I don't know why she talked about the biphasic release. I have no idea, because it just doesn't, other than if you're talking about intermediate like insulin, or a mix, it doesn't really make sense. So I don't know. This is what the body does. Endogenous insulin is released. There's a basal level, so meaning this kind of low level of insulin that's being released from your pancreas all day long, 24 hours a day, just kind of slowly being released. And then you have these like boluses of insulin that are being released right after your meals, and then going down, and again after your meal, and then going down, and again after your meal, and going down. And this is really what we're trying to mimic with insulins. I wouldn't worry. Well, if oh, I got a thing. Sorry, Darla's saying like the timing for when these islet cells, not antibodies, would increase, but the releasing of insulin. They would release insulin at these times, right? So while it would release like this low level insulin all day long, but then it's going to increase kind of in a bolus fashion when you eat, right? Because it wants to take up or use up as much of that glucose as it can. It wants to get as much of that glucose out of the blood as it can. It makes sense if you think it through. And this is what we base much of our like when we give insulin at the hospital, right? We give, most of the time, we're giving our short acting or our regular insulin, whatever, 20 minutes before a meal, because of this, because we know that when a person eats, the glucose is going to shoot up. If that person is unable to make or use their own insulin, they need to have insulin in the body when that happens. Otherwise, they're going to be hyperglycemic. There's going to be a lot of problems. So we give them insulin just before they eat to take care of that problem. And most of the people that I care for in the hospital will have a regular insulin, who are diabetic, will have their regular insulin before every meal, which makes the day really fun because you're running around trying to give trying to beat the meal tray to get your insulin out. Anyway, I just want to see where I was going next. There's so much in this. We did incretin hormones. Okay, so we can actually talk about. So we've kind of all like up until now. Oh, I did want to talk about this. Sorry, I should have gone over this when we talked about these incretin hormones. Again, remember I said these hormones, when you see the word incretin, it's always working on the side or on the side of insulin, right? So when you are full or you've eaten, you've got a lot of glucose in your blood, right? That upper GI and mid kind of GI, right? Your ileum and jejunum are going to release these two things that we talked about, the GLP and GIP. And I guess that's these little pink and purple things here. It's going to send a signal to the pancreas to increase your insulin and decrease your glucagon. So it's kind of the signaler of okay, we've got a lot of glucose in our blood, or we've eaten a lot of glucose. It's like another pathway for that message to be sent to the pancreas, I guess is what I mean, to release some insulin because we got a lot of sugar we just ate, right? A lot of glucose we just ate. And of course, when you release insulin or increase insulin and you decrease glucagon, then the liver is going to decrease glucose production, meaning it's going to stop breaking up glycogen, and it's also going to stop creating new glucose, and that way we get like a decrease in the glucose level in the blood. It's just kind of you can obviously, honestly, you can just kind of think, oh, okay, incretin hormones work with insulin. You don't necessarily need to know all of this, but it just makes sense to kind of go through it for me anyway. Okay, so all of that is just how the body works normally. Yay. Now we're going to actually talk about diabetes. So I'm sure you guys know a lot about diabetes already, but essentially it's a disorder of the carb and fat and protein metabolism. It results in or causes an imbalance between insulin supply and demand. Sorry, I'm just going to check. I heard a bing, so I want to check. What? Like the time? Yeah. Okay, you're good, Darla. Okay, so let's talk about like what those problems can look like. Your body can have an absolute insulin deficiency, meaning your body is not making any insulin. Nothing is being released from the pancreas. So you can kind of walk that through in your head and think like what's going to happen, right? You can have an impaired release. So your pancreas is making insulin, but it's not as much. The supply doesn't meet the demand, okay? Then you can also have an inadequate or defective insulin receptor. Basically, your cells are unable to actually use the insulin you have. So you could be creating enough insulin, your pancreas releasing the exact amount of insulin that you need, but because your insulin receptors are broken, they aren't going to be able to trigger this whole like opening up of the glucose channel, right? And sometimes you have insulin that kind of is just destroyed as it moves throughout the body. This one isn't as important. These three are kind of your big, most important kind of things that are happening behind the scenes, right? This is what's causing what's causing that diabetes. There are two main types. So I like that, Kieran, last year you guys did both of them in the same week. She split them up this year, so we're only going to talk about type 1 this week, and I'm assuming type 2 next week. But really, really quickly, because it's so important to know the difference. Type 1, you have this kind of abrupt onset. Usually it happens during childhood. Very few of diabetic cases are actually diabetes type 1. Most of them are diabetes type 2. really, for the most part, it's caused by little or no insulin production. So up here in this little list, it's either this one or this one with diabetes type 1. This here, this third one, where there's some kind of issue between the insulin and the receptor, the insulin receptor, is usually diabetes type 2. It's that, you know, we keep talking about insulin resistance and decrease in insulin sensitivity. That's kind of what we're talking about. This is resistant to the insulin, or it's not sensitive to the insulin, right? And that's usually in diabetes type 2. So this is not caused by, whereas diabetes type 2 is caused by or related to like obesity and like fat intake and so on. Type 1 is actually more of a genetic, they think more of a genetic or autoimmune issue. So it can actually happen to somebody whether they're like obese or thin. This is where you're going to get ketosis, and we'll talk about why in a bit. In diabetes type 1, insulin is always required. So a person who has diabetes type 1, they're not making enough insulin, or they're not making any insulin, they must have exogenous insulin. They have to have insulin given to them, or they will go into DKA and basically go into shock and die. And just I put these kind of little clues in everywhere. So if you have an absent or insufficient insulin, what's happening? Your glucose is lingering in the blood, and it's unable to enter the cells to be used in metabolism. So you can imagine what's going to happen to your body, right? Whereas in diabetes type 2, you have this like insulin resistance that's going on. So insulin can bind to the receptor, but it might not cause that cascade that's opening the glucose channel, or it can't bind at all. Or again, you can have where you don't have enough insulin made. But to be honest, it's usually one of these top 2 that's causing your diabetes type 2. We'll talk a lot more about that next week, so I'm not going to go too far into it. Let's talk about like what causes diabetes type 1. So this is, it's not a theory, like we know this is what's happening. It's just, it's not very well defined, I guess is what I'm saying. So people have some kind of genetic predisposition towards diabetes type 1. And then usually what happens is there's some kind of triggering event. More often than not, it's some kind of infection. That results in like the production of antibodies, and those antibodies eventually, or because of the acuteness, I guess, of the infection, those antibodies can turn on your beta cells. But that won't happen unless you have this genetic predisposition, right? So you have to have this some genetic predisposition. We don't really know where that gene is, right? So you have this triggering event. I think there are other things that can trigger it, but more often than not, it's the infection. Sometimes it can be something like MI, sometimes really increased stress, but like severe stress, where you have a lot of cortisol, a lot of epinephrine, a lot of glucocorticoids being released into your system, right? All of that can trigger kind of this predisposition that's already sitting there. So it basically leads to a decline of insulin secretion. The symptoms usually won't appear until about 80 to 90% of those beta cells are destroyed. So onset usually happens in childhood, but like not later in childhood. I usually, when I worked at Sick Kids, I did a placement at Sick Kids. Usually like 5, 6, 7-year-olds were coming in. And sometimes type 1 can develop in adults in their like late 30s, early 40s. It's pretty rare, but it does happen. So kind of keep an eye on that in case. Yeah. Oh, this was another thing. So you've got here, I was kind of, sorry, virus, bacteria. This was supposed to mean stress. And then actually toxins, like any kind of poison or something, could also trigger that genetic, this kind of destruction of beta cells. And what happens is the body creates antibodies. Of course, it's creating antibodies against that virus, but it's also creating antibodies against these, well, specifically against islet cells. So those are called islet cell antibodies, or ICAs. You know, basically these are destroyed over time. They're destroyed over time. This is the cause of that reduction, 89% of beta cell function, right? Because those antibodies are actually attacking the islet cells. They also attack this glutamate acid deoxycarbolase or whatever. It's just GAD65. So this is found in 80% of people with diabetes on presentation, diabetes 1. This is what they're saying is when they do blood work, they find antibodies to this GAD65. They find antibodies in the blood. Same thing with ICAs. They find those in the blood. Another place it attacks is insulin autoantibodies, or IAAs. You don't need to know a ton about this. It's actually really complicated, but you know, if you have, I think this is really neat that they can do tests now where if they detect ICAs and IAAs, so antibodies that are working against islet cells and against insulin, plus you have a decrease in insulin, they can predict that you will develop, type 1 diabetes within about 5 years. So in terms of this, I think understanding that diabetes type 1 is caused by this kind of destruction of these beta cells, and that it's an autoimmune response is really, really, really important. It's something has triggered an autoimmune response that is now, and it's destroying those beta cells. That's really for a test. I mean, it's really good to be able to recognize these GAD65, ICA, and IAAs as like signals that something is going on, that there's antibodies in the blood. But other than that, like, I wouldn't go through memorizing what each one of them does. There was a slide in your stuff that just talked about like why it's increasing, like kind of had like a theory as to why it's increasing. So if I were studying for this exam, I would just read through this and make sure I could recognize like if it showed up. So like I'm just gonna let you guys do that on your own. It's really not difficult. Let's talk about symptoms. So most common symptoms of diabetes type 1. So this is where things get a little bit complicated. We're going to talk about osmotic diuresis and stuff like that. So perk up, here we go. Polyuria. Poly, anytime you see that, lots of urea, we're talking about urine. So polyuria is a lot of urine. This is caused by osmotic diuresis secondary to hyperglycemia. What that means is when you have, let's say this is your blood vessel, you have a lot of glucose in your blood. Glucose is actually quite a larger molecule. Remember we talked about in our fluid electrolytes about albumin, and how because albumin is such a big molecule, it pulls water out of the interstitial spaces and out of the intracellular space. With glucose, something very, very similar happens. It changes the osmotic, like the gradient between these two kind of spaces, and you're going to have it's going to pull fluid out of your interstitial spaces, and it's going to cause, obviously that's going to contribute to an increase in urine output. There will also, we'll talk more about it later. But that's, I mean, just if you just think of it like that, you're good to go. Polydipsia, lots of dipsia and is thirst. So polydipsia is basically a response to your hyperosmolar state and dehydration. You're hyperosmolar because you have so many glucose molecules in your blood, right? It's hyper or above the norm osmoles, basically hyperosmolar, and dehydration because you've now had all of your fluids pulled out of your interstitial space and some even of your intercellular space, right? So now you're dehydrating and you're thirsty. Polyphagia, again, this is phagia with a G, so we're talking about eating or swallowing. Lots of eating or swallowing just basically means you're hungry, gives you hunger. This is like a hypothalamic response to the fact that you don't have glucose available. So even though there's glucose in the blood, you don't have insulin that's allowing that glucose to be used, and your body basically thinks, I don't have enough glucose. I need to get more in my system, and you feel hungry, right? It makes you want to eat so you can get more glucose. And then, yeah, you'll end up with malaise, muscle cramps, blurred vision. But these 3 Ps are really like your main symptoms, your main signs of diabetes. And it's insane. Like it really, somebody coming in with a new onset diabetes type 1 will have all of these things just textbook. It's kind of insane where they'll say like, I've been really thirsty the last 2 days. One of the things I always ask in Emerge, when you do a quick head to toe, always ask like, how are you peeing? Is it normal? How are your bowel movements? Like all of that. And usually they'll say, oh my gosh, I've been peeing like every half an hour and so much. And at that point, usually their lips are all dry and you can tell something major is going on. But we'll talk about that a bit later as well. How do we treat diabetes? Basically, insulin. Diabetes type 1 is always going to be insulin, and that's because they're unable to use any of their serum glucose on their own. So they're going to have we need to give them some way to do that. And if we do not, I'm going into ketoacidosis, diabetic ketoacidosis, and dying is definitely not an option, but something that may happen. So lifelong insulin therapy, we have to self-monitor blood glucose levels and then titrate insulin accordingly. You guys have probably all done this in your clinicals where, you do a lot of hospitals, you guys can't do it as students, but your preceptor would go and use this little doohickey down here to take their CBG, their capillary blood glucose, and depending on what that is, you probably have a sliding scale that tells you how much insulin you should give or shouldn't give. Usually it's at least 2. With diabetes type 1, you're usually giving a long-acting underneath, like to imitate that basal kind of slow release of insulin we talked about earlier, and then 2 or 3 throughout the day to imitate that kind of jump of insulin during meals. We'll talk a little bit more about that in a bit. So yeah, I think we've talked about everything here. I just want to make sure. Chronic hyperglycemia, so people who don't control their diabetes well, you have a risk of microvascular complications and cardiac events. Well, I think we'll talk about these more next week. These are things like your retinopathy. You have issues with nephropathy and neuropathy as well. I think we'll talk about them next week. Yeah. So let's talk about insulin. Again, I put this like 4 times. It's always required in type 1, sometimes in type 2. Many people that we see in the hospital end up being on insulin, who have type 2 diabetes are on insulin. And that's because the population who are in the hospital are people who haven't necessarily been compliant, right? And so they end up needing insulin as they get older. This insulin that we make, that we give them, I like how I say we, like I actually make it. It's identical to the human endogenous insulin. It's made from this like recombinant DNA technique. There are several different preparations available, and depending on the structure and the amino acid sequence of the actual insulin molecule, you could have a different onset, a different duration of action, and this is how we customize treatment for different people. This is important to know that exogenous or man-made or mad-made insulin can do all of the things regular insulin can. So it increases your fat, your carb and protein metabolism, it stores glucose in the liver, all of the same things. And it's important to note that it can't reverse any defect in insulin receptor sensitivity, meaning the diabetes type 2, usually that's caused by your body cells not being able to use insulin because there's a decrease in insulin sensitivity. That it's not going to change that. That's a permanent problem. What it can do is increase your insulin so that those cells that are working are able to take up more glucose. So this is super important. Obviously, I'm sure you guys know this. Definitely something you have to know for the NCLEX and for work. So you've got these kind of 4 classifications of insulin. You have your rapid acting, short acting, intermediate acting, and long acting. It might seem a bit overwhelming, but they're really not. You should be able to recognize them on the test. Let's compare like rapid acting and short acting, because those are really the 2 that people get mixed up. The others are easy. Lispro and Aspart are your 2 main rapid acting. You might see those are generic names. You might see the name brand, but they'll almost always have those generic names on them. There's also like the Solostar, I think it's like Admelog. Aspart is usually is Novorapid. Lispro is Humalog. And then there's this new stuff, this Fias, I've never used it, but it's actually extra fast. So I just kind of put it started over here. But so I had a question on NCLEX about insulin, and you just had to know like when. I think they said they were giving like Aspart, and then you had to know when it was going to kick in, basically. Your rapid and short acting insulins are always clear, meaning like the liquid that you're putting in, if you're drawing it up, it's a clear liquid, it's not cloudy. Your rapid acting is going to kick in within 15 minutes. It's going to peak at about 2 hours, and then it's going to last anywhere between 3.5 to 5 hours. What's important out of all of these 3 is your onset, really, and that's because, well, and your peak, but more onset. That's because we're giving insulin. When you give insulin, if the patient doesn't eat within those 15 minutes, usually we're giving rapid and short acting, before a meal. Honestly, usually it's more often than not the short acting, but anyway, we have to make sure the patient then eats. If we give insulin, we're going to drop their glucose, right? The insulin's going to trigger the glucose. It's going to leave the blood, and it's going to go into the cells and do all the things we talked about. So they could become hypoglycemic if they don't eat. So it's really, this is kind of the thing that shows up on tests a lot, is having to know when that patient needs to eat by, if you give them aspart or if you give them Humulin R, when are they at risk for hypoglycemia? Because that's really what we're worried about when we're talking about these insulins. So this kind of new kind of aspart here called Fiasp, this stuff is awesome. It kicks in 4 minutes, peaks in 1 to 2 hours. It's just basically the same. Only lasts like 30 minutes to 1.5 hours. So this is like when we need insulin to hit fast, right? 2 minutes before the start of the meal or within 20 minutes after, this is really the important thing that we need to know. So again, rapid acting, Lispro, aspart, it's clear. We give it at mealtime, and the effects can last up to 5 hours after the injection, so you want to keep an eye on the patient. Again, it's all about the risk of hypoglycemia. So if you're giving it at mealtime, make sure they're going to eat, especially if you've done their blood sugar and it's like 4.8 or 5 or whatever. Quite frankly, if it's that low, I'm probably not going to give it anyway. But you know what I mean, right? You got to keep an eye on that risk. Short acting, again, same thing. We're giving it at mealtime to kind of imitate that natural insulin release in the body. They always have regular or R in the name. So you're going to have regular insulin or Humulin R. It's always going to kind of be obvious that we're talking about regular insulin. I find, so that's really, really easy to remember, right? These intermediates always have like N or NPH. Novolin is an N, right? So you kind of know. I find the intermediates easy to recognize. The long actings are easy to recognize. Really, it's glargine or detamere. That's really all we're giving. I know there's a long list of a bunch of other ones, but these are the ones that are going to show up. Your Lantus and your Levemir. And then it's really just a matter of remembering, aspart and Lispro. I mean, it's really the only thing left, right? So if you see aspart or Lispro on a test, you know it's not regular because it doesn't have the word regular or an R in it. You know it's not intermediate because it's not N, NPH, or Novolin. And you know it's not Lantus or detamere. So it's got to be a rapid acting. And again, short acting. Again, this is your regular insulin. This is the stuff we're giving. If the patient is in DKA and we give IV insulin, this is what we're giving. We're also using it before meals. You can actually combine this short-acting regular insulin with an intermediate acting insulin to kind of, everybody's different, right? And we want to try to get them as close as we can to imitating this kind of endogenous bolus pattern. So onset now, instead of 15, it's 30 minutes. It peaks in 2 to 3 hours and lasts 6.5 to 7 hours. Again, these are pretty, this is like the norm, right? So this is the regular insulin. This is the norm. So I kind of always memorize this first and know that it's about 30 minutes. And then I just I know that if it's aspart, it's going to be a lot quicker. And these intermediates and long actings are really easy to recognize because they're really long acting, like 18 hours or 24 hours. You're never going to see a regular insulin that's going to last that long, 7-8 hours tops, right? So again, I'm trying to give you my hints instead of just, you guys know this stuff. It's just having to memorize it to pass a test, right? So again, your intermediate is going to be Humulin N, NPH, Novolin. This is your cloudy one, okay, that cloudy. Onset in 1 to 3 hours, peaks 4 to 10 hours, up to 18 hours duration. Again, these numbers are quite big. You know when you see these, it's not going to be a short acting or a regular acting, because your short and regular are going to kick in within 30 minutes tops, right? If it takes 1 to 3 hours or an hour and a half to kick in, it's got to be intermediate or long acting. This is the stuff we're giving to kind of have that basal rate that's going underneath. And again, we can combine it with this regular acting. We can mix it together to kind of tailor it to the patient's needs. And again, long acting. So then long acting is another kind of basal, right? That's keeping that rate coming out a little bit all day. Again, most popular glargine, which is the name brand is Lantus, or Detamir, the name brand is Levemir. This one is clear as well. The only one that's cloudy is your NPH, which is your intermediate. Onset in 90 minutes. Again, you see that, you're going to know it's not kicking in within 30 minutes. So it's a long acting. There's no peak because it just literally flows like this all day, and it lasts 24 hours. So if you're asked a question, which most likely the question will be framed something like, you're given this, you've given your patient this, and they'll give you the name of it. What do you expect to happen, or when should the patient eat, or what have you? So you just kind of have to be able to recognize. You don't have to memorize. The long acting, we give it Q12, so bedtime and in the morning, or sometimes we'll only give it once a day. And in the hospital, it's almost always given with the short acting as well, so that you have that basal rate going underneath, and then you also have that bolus happening with the regular or the short acting. All you need to worry about when you're talking about mixing insulins, we only mix regular and intermediate, and sometimes they come pre-mixed. For example, you can get this Nova mix that's 30-70. It's 30% aspart, which again is rapid. Sorry, I must have mixed that up. Anyway, sorry. So let's start at the top. You got 30-70 is 30% regular insulin, 70% NPH. So this is regular and this is intermediate, right? And they all are kind of like that. There's nothing really important. This is where that biphasic comes into play. Gives more of a biphasic result. So you can kind of match that up with whatever Shireen had kind of talked to you guys about the biphasic. Like I said, I don't really think that way at work, because we do much, yeah. Anyway, this is like more, if you're working in a diabetic clinic, this is definitely stuff you have to know. When you're hand mixing your insulin, you have to draw up the regular insulin first. And if you guys remember from any of your practical tests or whatever, it's cloudy, clear, clear, cloudy, right? And so you inject the air into the cloudy, then you inject the air into the clear, then you pull up the clear and then you pull up the cloudy. So you can just look at this and just do whatever you have to do to try to remember that. That often will show up on a test. Make sure you understand what a sliding scale is. If you look here, and this is really, really common, it'll tell you when you go to the patient at a certain time of day, you take their CBG and it's 12.1. It tells you how many units you need to give them. And this is like a directive, so you don't have to get an order or anything. You just grab the pen and do it. Where are we going here? Okay, so I think you guys know already all of this stuff. It comes in a pen, right? You can't take insulin orally. It has to be sub-Q. Sub-Q is the most common way. IV, we really just do in emergencies. Want to have it in a 90-degree angle unless the patient is really, really thin. Then when you pinch, the basic idea is that you want to get it into this sub-Q space here, not like in the just under the epidermis right here, because then it's going to just kind of go everywhere instead of hitting that sub-Q space. That's where we want it. So if you have a really thin person, you have to pinch and maybe go in at a 45, but usually 90 is fine. Insulin pens are awesome. They click as you pull it up. So if you turn the top, for example, and you have to give 10 units. You turn it 10 times, like click, 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 click 10 times. There's usually about 300 units in it. You change the needle. That's a really important thing for a test. You change the needle every time with every injection. You never, ever, ever use the same needle more than once. They come in little tiny packages that you attach to the pen. You reuse the pen. The Basil infusion pump, this is amazing. I have a friend who actually has one of these, and it just changed her life. It's a battery operated kind of little pack. It's about the size of a pager. I mean, they used to be a lot bigger. And you have this kind of little sub-Q sticker that with the little sub-Q needle that's kind of on you all the time. You do have to like clean it, whatever, but it's attached through this cannula into this little battery operated pack, and inside of this pack is this big thing of like a reservoir kind of insulin, and it mimics that basal rate, kind of lets it go through all day, and then you program it based on kind of your typical blood sugars. You would program it to release that kind of more bolus throughout the day. Have to be really careful with these pumps that you're eating regularly, and you kind of keep a very, very like a similar pattern of eating every day. Obviously, there are issues with it's battery operated, so you want to make sure it doesn't run out of batteries. Patients have to learn how to program it, so it can be a little bit difficult with that. What's really important with these, and this is the question that comes up in NCLEX prep a lot, you still have to monitor your glucose regularly. You can't just, depend on that. You could go into hypoglycemic shock very, very quickly if you're not paying attention. And again, you need to change the tubing every couple of days. We always anybody who's taking insulin, we have to teach them to recognize the signs and symptoms of hypoglycemia. They're here. Your main ones are clammy cold or sweating, this lack of energy or fatigue, hunger, and tachycardia. Those are like your main four kind of that everyone, like your textbook ones. And then depending on the severity, you treat like based on that, right? So if it's a mild hypoglycemia, mostly autonomic symptoms like trembling, palpitations, sweating, and so on, you usually can treat yourself, drink some juice, have a cookie, whatever. Same with moderate. But when it gets severe, you actually will have, my friend that's on the basal pump had severe hypoglycemia one day, and her husband had to call an ambulance. It was super, super low. She just was completely unconscious and unresponsive, so it was terrifying. But that's when it gets really, really low, and this is why we constantly are monitoring your CBG. Okay, whew. That's the basics. We got one more thing we have to talk about, which is the diabetic ketoacidosis. I just need to take like a 2-minute break, and then I will come back and we'll go through it. Hopefully you guys are still around. You're sticking with me. We have, I'm hoping I can get through it in about 15 minutes, and then we're done for the day. So I'll be back in just a couple of minutes. Just give me a second. Okay, I'm back. Hopefully everyone's still with me. I know this is huge and it's a lot, but let's talk really fast about DKA. The This is the really serious complication of type 1 diabetes. Severe insulin deficiency. Basically, it's an absence of insulin. Patients develop glycosuria, which is basically glucose in your urine, severe hyperglycemia. Some patients come in and they're so hot, they're so hyperglycemic that it doesn't even show up on the finger stick, like it malfunctions. Yeah, they're going to have increased lipolysis. That makes sense, right? And then increased ketone production. Again, with the Y. I got to fix that. And then the increase in these ketones, you remember when you break down that fat, you're going to release those ketones. The increase in that ketone causes ketosis, and the increase, this is important, that the increase in the ketone causes an increase in the hydrogen load, hydrogen ion load, right? And that's where you end up with metabolic acidosis. For me, I just, because it's called ketoacidosis, I know that we're talking about metabolic acidosis and not alkalosis, right? It's super easy. So what can kind of trigger this? Because we're talking about an absolute or almost absolute insulin deficiency. There's no insulin. How does that happen? Usually it's the onset of diabetes type 1. Patient doesn't know they have it, and something has triggered it. Most of the time, again, it's an infection. You have an increase in those glucocorticoids, an increase in gluconeogenesis. Because of that, you have an increase of glycogenesis in the liver. Basically, what you have is that hoarding, again, of that glucose in your blood, and you end up with this very high, really high glucose levels in the blood. Body, if they have any insulin at all, the body uses it up very quickly, and you end up with this deficit. Sometimes this can happen in a patient who is diabetic, knows they're diabetic, and is non-compliant with their medications. Sometimes it can also be triggered by some of these other things, right? Your MI, trauma, all of these things will increase the glucose level in your blood, right? Cushing's disease specifically has an increase in cortisol and glucocorticoids. I mean, that's what Cushing's is. Again, here's that severe stress, because you have that SNS response. It's going to bring out all of this stuff again, right? This epinephrine. And we know that epinephrine is a counter-regulatory hormone. So we'll go through this quickly. So what does the body do? You have no insulin. So there's no insulin to suppress that lipolysis. So remember we talked about what insulin does, and we don't need that fat to be broken down, right? So one of insulin's job is to decrease that, or to stop that, make it slow down, stop it from happening. When you don't have insulin in your body, you're going to end up with an increase in lipolysis. And that's despite the fact that you actually have a lot of glucose. So usually what you see is your body starts to break down fats because you don't have a lot of glucose. In this case, you have a ton of glucose and your body's breaking down fat still. And that's because there's no insulin in your body to stop that from happening. And so your body begins producing, part of the reason, as well, is that you have a ton of glucose in your system, in your blood, but none of it is able to be used to make energy, because the insulin is what gets it into your muscle cells and your fat cells, and it's not getting in there because there's no insulin. And so your body, again, will start to break down fats. It needs to get the energy somewhere, right? When you break down fats, fatty acids are released. And you end up with ketogenesis, which is, again, genesis, the beginning of, or the creation of, ketones. When this ketogenesis occurs, you end up creating this acetyl-CoA, which is, it's the first step of that Krebs cycle. Remember earlier we said cellular, in order to make those 34 ATP, we start with glucose and oxygen and we end up going through those three kind of different levels, the glycolysis and the Krebs cycle. That this is the second step. We need this acetyl-CoA. So we can get that by breaking down fat and causing ketogenesis if we don't have enough, glucose. Obviously, once we do that, we're going to have a bunch of ketone bodies released into the blood and into and that's going to get filtered through the kidneys, and it's going to show up in our urine. So one of the main symptoms of or signs of diabetic ketoacidosis is ketonuria. In other words, there's ketones in the urine. Again, those ketones are acidic, so we are could cause a metabolic acidosis, not always. But you're going to end up with a pH less than 7.35, an increase in your anion gap. Your anion gap is just measuring kind of the relationship between the positive and negative ions in the body. If you look over here, I put all the norms in for you so you don't have to get lost. These are the norms that matter in DKA. Your keto acids are, building up. One of the things keto acids do is they consume or they decrease bicarb. If you remember from your acid-base balances, bicarb is base. So it's what's working against acidosis, and now we're decreasing it. So we're worsening that acidosis, right? Once you get metabolic acidosis, you've got all kinds of symptoms. You've got this disruption in your enteric nervous system, that's your GI system, which is going to decrease gastric emptying, could cause ileus, which is basically a paralyzing or peristalsis, complete stopping of the GI system. And that's why you end up with this abdominal pain, nausea, vomiting, and this is going to increase dehydration as well. But we'll talk about dehydration in a second. Again, the metabolic acidosis, the body is going to compensate. It's going to try to compensate. And if you remember from your acid-base balance, the carbon dioxide can increase acidosis in the body. There's anyway, and so the body can compensate for acidosis by trying to blow off some of that CO2. How does it do that? It takes really, really deep breaths in and blows really, really deep breaths out, trying to get rid of that CO2. And that exactly pretty much describes your Kussmaul respirations, right? Your deep, fast breathing, because they're trying to blow off the CO2. Your body will also rid itself of some of those ketones by blowing them off or excreting them through your lungs. And this is one of the big, big signs of diabetic ketoacidosis, and that's acetone or fruity breath. This is so it really does, it's very sweet smelling. You can catch it almost right away. And then again, with your metabolic acidosis, you're going to have a disruption in the signaling to your brain, spinal cord, and nerves that's going to cause some more neuro symptoms like weakness and confusion, and can increase into a coma. Sorry, you guys, I'm getting really tired now. Okay, so that all kind of happened from this absolute insulin deficit. What else will your body do? You have no insulin. So your glucose, you have tons of glucose. It's staying in your blood. It's not being used by muscles and fat cells because those muscles and fat cells need insulin, right, to open that GLUT4 receptor. So that immediately is going to trigger the hypothalamus. It's going to sense that low intracellular glucose. It senses the fact that there isn't glucose inside of the cells, even though there's tons of glucose in the blood. And that's going to trigger that feeling of hunger or the polyphagia. So you're going to get crazy, crazy polyphagia, sorry. Also, glucose remains in the blood. It's not being used. So you're going to have, that's going to contribute to hyperglycemia. And we're talking hyperglycemia, like very high. The cells themselves are not getting enough glucose, right? And they're starved of glucose. So the body thinks it doesn't have enough glucose, and it triggers the release of all those counter-regulatory hormones to try to to try to increase the glucose level in the body, even though we already have too much glucose. And so those are your counter-regulatories. We talked about them already. The body's trying to increase the blood glucose, hoping to increase the cellular glucose absorption. And again, that's going to decrease your protein synthesis, meaning it's going to stop your muscles and from breaking down protein anyway. Sorry, I'm really getting fried. And then you're going to increase, the important thing is this, it's going to increase your gluconeogenesis and glycogenolysis in the liver. So your liver is going to start to make new glucose, and it's going to start breaking down glycogen to release more glucose into the blood. So that is going to contribute to even more hyperglycemia. That hyperglycemia itself, from that increase in gluconeogenesis and is going to result in an increase in glucose. It is going to be very, very high. I don't know why this says 12. I might have copied it from somewhere else, but it's usually much, much higher than that. Normally, the kidneys reabsorb almost all of the glucose that goes through them. So the blood carries the glucose to the kidney, it filters through, and most of it is reabsorbed, I think in the proximal convoluted tubule. So it's reabsorbed. But when you have this much glucose in your system, your kidneys can only reabsorb so much, and you actually end up having glucose in your urine, or glycosuria. You shouldn't have glucose in your urine. So that's a huge sign that something that usually have DKA or very, very high glucose. That increase in glucose, right? We talked about how it increases changes the osmotic gradient, right? It's pulling all that fluid out of the interstitial space. So that's called osmotic diuresis. That's going to increase your urine output because you've got all that fluid that you're pulling out into your blood. It's getting filtered in your kidney, and then it's coming out as urine. So that's where your polyuria is coming from. And then it does trigger this severe dehydration. Like I had a guy come in with DKA once who, I mean, his lips were just cracked and dry, and his blood sugar was so high it didn't even show up on the CBG monitor. Sorry. So you've got this severe dehydration that's going to cause neural problems. So it's going to contribute again to that weakness and confusion and possible coma. And it's also going to alter the body, the total body water and ion osmosis, meaning those hydrogen ions are going to start jumping into the cells. And when hydrogen ions jump into the cells, potassium ions jump out. This is a really, really important concept of potassium and how it relates to DKA. So when you have this electrolyte imbalance, right? In general, you've got all of this fluid flushing out of your body. It's pulling right there. You're pulling out some of your sodium, potassium. So in DKA, you very, very often will have hyponatremia. You'll lose magnesium. You'll have a loss of a lot of different electrolytes. But there's kind of three big things that are happening with potassium specifically. One of them is that normally insulin pushes potassium into the cell. When you have a decrease of insulin, that potassium isn't being pushed into the cell and it's staying in the blood. It's being filtered and then excreted out through the kidneys. The second thing that's happening is that osmotic diuresis, that glucose, that large molecules drawing the fluid out of the cells without the insulin to push that potassium back in, your serum potassium is going to go up, right? And the third thing is your sodium and hydrogen are rushing into the cells. They're trying to balance out that osmotic gradient. So you have a lot of sodium, potassium, what have you. So inside of the cells compared to outside, because you have so much fluid now outside of the cells in that intravascular space. You've pulled it out of the cells. So the ratio of sodium, hydrogen, whatever, to water is so high inside of the cells that your body is trying to kind of balance that out by pushing hydrogen and sodium, into the cells. And whenever sodium and hydrogen go into the cells, potassium is going to come out, always. So so you end up with originally a lot of potassium outside. So when you pull potassium, you pull blood at the beginning of this kind of DKA workup, you may see the patient's potassium levels are normal or even high. They might look like they're hyperkalemic. And it's so crazy because the third thing you're going to do when you treat DKA is you're going to give potassium. And it's crazy because you're giving potassium sometimes when the patient's potassium is completely normal. But that's because it's normal because all that potassium has been pulled out into the blood and it's about to be excreted through the kidneys. It's about to all leave through urine. And so you need to prevent that hypokalemia from happening by giving potassium. So anytime your potassium is less than 5, you're going to give potassium. And yes, you want to ensure good renal function and urine output first so you don't end up with hyperkalemia, right? You've got all these symptoms of dehydration. You guys already know, one of which is polydipsia. And that's so that kind of highlights like all of the major symptoms of diabetic ketoacidosis. How do we manage it? The main thing that you need to remember, okay, always, you know this already, always ABCs first. So you're going to do, oxygen if they need it, whatever. But you always do fluids before insulin. This comes up a lot. People miss it a lot. You're always going to do fluids first. It feels like you should do insulin first because the reason you need to treat the hyperglycemia, but no, you need to treat the dehydration first. It's the more immediate threat. So if that shows up on a test, don't get it wrong. Fluids first. We usually use normal saline. Usually discontinue when the patient starts to drink, actually, but sure, when it's 30 milliliters per hour. Then we're going to do IV insulin. This is one of those times when it's an emergency. So we'll do a bolus of insulin and then we'll hang a continuous infusion as well. And there's your potassium. We're going to treat the potential hypokalemia. If it's less than 5, we treat it. And then the bicarb, remember we said that bicarb is getting absorbed or kind of kicked out by ketones. We may end up in acidosis if the patient, well, you're in acidosis, if you have ketoacidosis, you're in acidosis. If it's, I guess for textbook purposes, if it's below 7, your pH is below 7, you're going to give bicarb to treat that metabolic acidosis. And then you're looking at like other complications and other patient teaching and so on. The really important takeaway from this is fluids, then IV. And also you're giving potassium even if their potassium is like a high normal or normal at all, because it's going to deplete. I kind of did this little list over here so that you can see kind of just what I just said, what you treat first. And then your ongoing monitoring is pretty self-explanatory. You're going to do vitals, level of consciousness, cardiac rhythm. They're going to be on tele. You're going to leave an O2 sat on their finger. You're going to put in a catheter because you want to have an exact urine output. This is something that shows up on tests. You want to make sure that you're listening to breath sounds or monitoring breath sounds because you're giving all that fluid. You want to make sure you don't hear any crackles, you don't have any fluid overload. And then of course you're doing blood work pretty much every hour. You're going to do glucose, potassium, and pH level. Okay. So that was insane. That was a ton of information. I don't even know if everybody is still here with me. Probably you have all fallen asleep.
No, we are here. Thank you.
Syed, of course you're here. Anyway, I'm exhausted. Does anybody have any questions? Oof. If you're still here and you have questions, feel free. I'm just gonna give you a second. Kieran, are you still here?
Yeah, I'm here.
Stay awake for all that? Oh my gosh, it's so long. Okay, so I don't think you guys have questions just because you're in overload right now. That was a lot of information, so just kind of go back over the video one piece at a time before you study for your test. I think I highlighted like the things you really have to know, even though there's a lot of information. It's just the really, really like particular things you have to know for your test. So you should be okay. I'm just going to click off the recording.